Hello, everyone, and welcome to today's webinar titled Developing Methods for Comparability Studies of Therapeutic Monoclonal Antibodies. Minimize time, maintain quality. This webinar is part of the 6th Annual Bioprocessing Virtual Event Series 2024. I am Helge Schneer, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and sponsored by Sartorius. For more information about Sartorius, go to www.sartorius.com. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to first participate by communicating with other attendees using our live chat feature during the presentation. You can find the live chat located at the left of your screen. You can also participate by submitting as many questions as you would like during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We will answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Help Desk button located at the top of your screen within the navigation bar or from the lobby within the event. Now, let's get started. I now present today's speaker, Kalhari Silva, Head of Scientific Innovations and Research at Custom Biologics. For a complete biography on our speaker, please visit the Presenter tab from the menu of the left of your screen. Kalhari, you may now begin your presentation.
It's my pleasure to be here today. The focus of today's talk will be how we at Custom Biologics develop methods to perform comparability assessments of therapeutic monoclonal antibodies, while we also ensure we use our time wisely, while we maintain scientific integrity as well as quality. I am Kalhari Silva, Head of Scientific Innovation and Research at Custom Biologics, where I provide technical oversight to scientific development and validation activities for both bioassay and molecular diagnostic divisions. My team is composed of about 15 scientists, very talented, hardworking, who have joined us from all parts of the globe. Custom Biologics is a Health Canada audited, GMP-compliant global service provider for functional testing of biotherapeutics, biosimilars, as well as antibody drug conjugates. Custom Biologics was established in 2004, and all work at Custom Biologics is conducted in compliance with Health Canada CGMP drug establishment license, as well as in compliance with Public Health Agency of Canada Pathogen and Toxin License. We have two main divisions that fall under the Scientific Innovation and Research umbrella, which is the Large Molecule or the Bioassay group and the Molecular Diagnostic group. So in the Large Molecule division, we are involved in the development and validation of ligand binding assays. That's where the octet comes in, and cell-based bioassays. They measure the biomarker profiles and expression levels, as well as the binding, functional activity, and immunogenicity of various biologics, monoclonal antibodies, and biosimilars. In other words, it's any therapeutic with a peptide backbone. On the other hand, Molecular Diagnostic Division uses highly sensitive and rapid nucleic acid amplification and identification assays in many areas of drug biomarker discovery, biopharmaceutical drug development, and also lot release testing. We are also currently involved in the development and validation of a molecular diagnostic test for the detection of SARS-CoV-2. So our mission at Custom Biologics is to basically employ established laboratory and statistical methods to conduct potency, comparability, stability, and lot release testing of biosimilars, not only in the course of research and development programs, but also as validated GMP-compliant assays that are suitable for stability analysis, clinical trials, as well as clinical lot release testing. So, monoclonal antibodies are one of the most widely studied molecules at Custom Biologics. So, with the development or with the introduction of the hybridoma technique in around 1975, that has made it possible to produce monoclonal antibodies in large amounts. And around the world, there are at least about 570 therapeutic monoclonal antibodies that have been studied in clinical trials by commercial companies. And 79 of them are approved by the US FDA. And 30 of them are actually used in the treatment of cancer. So, what are these monoclonal antibodies? So, the vast majority of marketed monoclonal antibodies, they belong to the IgG class, or precisely IgG1. And to some extent, there's also IgG2 as well as IgG4. So with more than 1,300 amino acids resulting in a mass of approximately 150 kilodaltons, these MABs or monoclonal antibodies are large molecules. They are built from two heavy chains shown in blue and then two light chains shown in yellow. Which are 25 kilodaltons in size, and the heavy chains composed of about 50 kilodaltons. So the different heavy chains divide these IgG into various subclasses ranging from IgG1 to IgG4, and the light chains can be of lambda type or kappa type. Heavy chains and light chains are also stabilized with the presence of inter and intra-chain disulfide bridges that hold the antibody in a Y-shaped structure. The heavy and the light chain also consist of different domains, and the variable domain as well as the glycosylation sites that allow glycosylation in the Fc region. And so the size and structure of the IgGs, they give rise to a large number of possible alterations and modifications. And these modifications or alterations have a tremendous influence, not only on the structure of the monoclonal antibody, but also on the pharmacokinetics as well as the function of the monoclonal antibodies. And our focus on today's talk will be how we use the octet to evaluate the impact of these alterations in the MAB's functional performance. So as we know now, MABs are complex structures and they also have numerous critical quality attributes. So what is a critical quality attribute? So if you look at the definition, it's a physical, chemical, biological, or microbiological property or characteristic that should be within an appropriate limit, range, or distribution to ensure the desired product quality. Biotech therapeutics, particularly complex products like monoclonal antibodies, they can have numerous quality attributes, and these affect the safety and efficacy of the product. So identifying these CQAs is supposed to be the first, but also arguably the most difficult in the development and production of biopharmaceuticals. This is where we have implemented the use of octet to assist many of our clients in the drug development life cycle. So for the talk today, I would like to refer to three case studies. The three case studies will look into how we optimize the binding assays of monoclonal antibodies, one to the FcR receptors, two to the macrophage mannose receptor, and three a more functional VEGF factor, vascular endothelial growth factor. So moving to case study one. So here the client consisted of three different antibodies, or it's a cocktail of three antibodies, which was generated from one expression system. So as the client migrated from one expression system to another, we were supposed to identify the comparability or how the binding changes of the three antibodies from one expression system to another in terms of binding to the Fc receptors. So we had to evaluate the binding of these antibodies to the whole Fc panel. But for today's talk, we will look into how we optimize the binding assays to Fc gamma R3A and Fc RN. So why is Fc gamma R3A so important? So this is because of the induction of a phenomenon known as antibody-dependent cellular cytotoxicity. So ADCC is a mechanism of action of antibodies through which virus-infected or diseased cells are targeted for the destruction by components of the cell-mediated immune system, for example, the natural killer cells, here depicted as the effector cells. So what happens is once the antibody binds to the target antigens on the cell surface, the Fc portion of this antibody then bind to the Fc receptors that are present on the cell surface of effector cells. And there can be multiple cross-linking that is happening between the two cell types, leading to a pathway activation of ADCC mechanism of action. And ADCC is then induction is then considered an important critical quality attribute and must be evaluated to ensure biotherapeutic efficacy. So now let's look into how a typical sensogram looks like. BLI measures the shift in reflected white light upon changes in the thickness of the biolayer. So the measured shift depends not only on the size, but also on the affinity of the interactors. So there's an initial baselining of the sensogram that happens when the light signal of in a buffer of choice, which is then followed by a loading, for example, onto the surface of streptavidin with a biotinylated ligand. And then again, this is baseline by incubating the biolayer back in our buffer of choice before being transferred to analytes in a series of concentrations free in solution. And then dissociation of the complex is then measured by transferring the biolayer back into the buffer. So when we optimize these BLI experiments at Custom Biologics, the first assessment we make is to determine if there's non-specific binding of the analyte to the empty biosensors. So if you recall the antibody cocktail study, here MAB1 and MAB4 represents two antibodies coming from the two expression systems. And here I have also used two different buffers, PBS and also kinetics buffer. Kinetics buffer is used here at 1x, which is diluted from a 10x kinetics buffer provided by Sartorius. You can see like a positive shift or a positive BLI binding signal of MAB1 and MAB4 in the presence of a buffer that is PBS, but that is considerably lower when the analytes are in a buffer such as kinetics buffer. But you also see a negative drift of the signals in the presence of PBS or kinetics buffer, which we do not exactly know why. So we have to avoid this negative shift or negative drift. So in that case, we needed to perform a load step before that. So to continue our optimization assays further, we use this nickel NTA biosensors. The nickel NTA biosensors was preferred in this FCR study because we can use FC receptors that are commercially available and consist of a his tag. So we do not need to modify any of our binding partners, neither the receptors nor the analyte or nor the antibody, and we can just immobilize the his tagged FC receptor onto nickel NTA biosensors. So here we can see, what we can see is in all the three sensograms, the blue trace represents the one with a load, and the magenta trace represents the one with no load. So when a sensor is loaded or not loaded, the difference that you see in the signal of a MAB is actually not that different. So this really suggests that the MAB is actually maybe binding just to the empty sensor and not to the load, or we do not even know what's exactly happening there. So then we wanted to move into different blocking reagents. That's where we started pursuing skimmed milk. So we used skimmed milk and we performed a blocking step, but most probably that caused the quenching of the signal, and we do not see any signal at all. And we proceeded to reducing the skim milk concentration, but still we did not see a very high differential in terms of binding versus no binding. So we have to continue our optimization experiments, and then we use different blocking agents. We continued using different blocking agents. So here what we see is sensor C, where we use 0.1% BSA as our blocking reagent, and sensor D, which is in magenta, is twin 20 at 0.05%, and then we have another sensor without any block. So here, all the three sensors are loaded. So we do not have a no load in this case, but you see that in the presence of the load, there's actually no negative drift. And the presence of 2020 actually gave a marginally better signal to noise ratio. So we used 0.1 2020 as our blocking conditions in the future. So we have another sensor which is loaded, as you can see from the red trace in the next sensorgram, and then we have another sensor without a load as seen by the blue trace. So here you can see that the signal is actually quite high for the loaded sensor in comparison to the one without load, suggesting that we have reduced the non-specific binding and we have obtained a good signal to noise ratio. So then our next task was actually to move on to the next pair of monoclonal antibodies. So this is the second antibody coming from the next one expression system and another antibody coming from the another expression system. So when we're performing comparability studies, it's always nice or it's always necessary to make sure that the comparability conditions are the same throughout so that you can make accurate estimations. So here we continue to use 2020 as our blocking. That is in the final sensorgram, but you do not see a differential in terms of binding of the MAP2 and MAP5 to a loaded versus no load sensors. So we're back to square one, and this all suggests that it's just non-specific binding. So we had to change our format a little bit. So that's where we moved on to a FAB2G biosensors. So this orientation here is a little bit different, because in the nickel NTA biosensors, we are using the His tag of Fc receptors. So there it is an Fc down approach, or antibody is free in solution. But here we are using FAB2G biosensors, which immobilizes the antibody via the CH1 domain of the Fab fragment. So it is called as the antibody down approach. So we load the MABs with the MAB here at 5 micrograms per ml, 10 micrograms per ml MAB, and then dip it into and allow the association of into Fc gamma R3A at 6.25 micrograms. But you see here, as depicted by the red arrow, that there's really no clear differentiation between a load versus a no load, suggesting that it's not worth pursuing it. But one might wonder, why didn't we increase the concentration of the analyte beyond 6.25 micrograms per ml? This was nothing but because Fc gamma R3A is a commercially supplied product, so it was becoming economically non-viable to pursue this approach, because we needed to buy a lot more quantity to proceed further. So we had to change the assay format and use SAX biosensors next to determine if either the receptor down or an antibody down approach works. So here in slide 12, we can see the assay optimization with SAX biosensors. SAX biosensors are high precision streptavidin biosensors, which immobilize the ligand via a biotinylation process. So to fast track our optimization efforts and to save on reagents and on our efforts as well, we did not invest time on biotinylation first, because we didn't know whether this approach would be taken or not. So initially, we wanted to see the non-specific binding of all the 6 MAPs, 3 arising from each cocktail, to an empty SAX biosensors. So we use the throughput capabilities of the octet. We use 8 different sensors, 6 of them dipped in 0.5 mix per ml MAP, each MAP, and then 1 dipped in 1x kinetic buffer, and another also dipped in Fc gamma R3A. So mind you, none of these are tagged, or none of these are biotinylated. It's just to see if there's non-specific binding of the monoclonal antibodies and the receptor to the empty biosensors. So here we can see that the maps arising from one expression system, for example, MAP1, MAP3, MAP5, has more non-specific binding in comparison to MAP2, MAP4, and MAP6. But if you look at the BLI response signal, the binding in nanometers that you see on the left side is very, very low. It's even lower than 0.1 nanometers. So this was promising. And then we wanted to biotinylate the receptor and use a higher load to see if we could amplify the signal and also reduce the MABs further to overcome the NSP. So it was a two-way approach. So here, this was the last experiment we did for the method establishment, and we really like to see this. So here, what we did was we used 6 biosensors. 7 biosensors were used in one assay, and 7 biosensors were used in one assay. So this was actually not 2 different assays, but they were used in parallel. So it was 2 different sets of 7 biosensors that were run in one particular assay to see the non-specific binding. So the no-load biosensors, those are the sensors without any load, were dipped into the same MABs, and the ones with the load of biotinylated Fc gamma R3A, 5 micrograms per mil, were also dipped into the MABs. So as you can see, the no-load biosensors were literally flat in the association stage, despite being dipped into the same MAB solution. And the ones with the load had a very nice BLI measurable signal, suggesting that it is it's not an artifact, but it is a specific binding event. So having established the binding conditions, we then moved on to our final assessments. To obtain the reliable and accurate kinetic constants, it is recommended to use a dilution series of at least 4 analyte concentrations. So here we used 4 different analyte concentrations in our association step. To account for assay drift, the measured BLI response of the reference sample biosensor, that is a sensor dip already loaded but dipped in 0 analyte concentration, was subtracted from each of these measured BLI responses. And the data was analyzed globally by fitting both association and dissociation phases of several analyzed concentrations simultaneously to a 1-to-1 Lagmuir binding model, resulting in the generation of affinity constants, on rates, and off rates. And as per the requirement of our client, we had to do independent triplicate binding study on 2 different days. And as you can see, the binding constant generated are quite reproducible and reliable based on the values in general and the mean value, the average value, and also based on the standard deviation. Look into the next receptor. This is the FcRn, or the neonatal receptor. As you know now that the Fc region of human IgG, they contribute to a number of beneficial biological as well as pharmacological characteristics, such as the one that we talked about just now, the ADCC. Another one is that is most important is prolonging the plasma half-life via its unique pH-dependent interaction with the FcRn, or the neonatal receptor. So IgG half-life is primarily regulated by binding to this neonatal receptor, FcRn, through the IgGs, or the MABs constant region, the Fc portion in particular. So what happens is that once the Fc and the FcRn binds in an acidified endosome around pH 6, it gets rescued from the cellular degradation pathways you see in red in color in this case. And then the intact IgG is then released back into the bloodstream at neutral pH, or physiological pH of about pH 7.4. So this altered FcRn binding can either increase or decrease the serum half-life of this Fc-containing therapeutics, and that in turn affects the drug efficacy. So hence, FcRn binding interactions are increasingly being assessed at multiple stages of biologic drug development. So to understand the pH-dependent binding of FcRn and MAB, we performed a pH-dependent type of experiment. So in slide 16, what we see is the pH-dependent binding of FcRn and monoclonal antibody binding. So here, 7 biosensors in total are immobilized with 5 micrograms per mil of the neonatal receptor. which is biotinylated. The biosensors are then transferred to kinetics buffer, which is indicated shown in the baseline at different pHs, ranging from pH 6 all the way to pH 7.4. And then these biosensors are transferred to the analyte that was also prepared at different pHs before being transferred back into the dissociation phase. As you can see, the association, as you can see on the sensogram on your left side, the association increases with decreases in pH, being the highest at pH 6 and lowest at pH 7.2. And on the right shows a binding isotherm that is similar to a physiological condition where the map binding to FcRn occurs at pH 6. It depicts shown by the blue line, and where it is released to the bloodstream at pH 7.4 quite fast or quite quickly. So similar to our previous method establishment, we also did a similar scenario to FcRn, where we use two sets of parallel biosensors, 7 each, loaded and without a load. And you can see that the non-specific binding is minimal when the no-load biosensors are dipped into the same map preparations, and the signals are quite high in the presence of a load, suggesting the presence of a specific binding event. So this is a snapshot how a data set looks like when the binding interactions were studied at pH 6. It's a triplicate binding study again, performed on 2 different days. And as you can see, the binding constants are very close to one another. They're highly reproducible and reliable, as indicated by the mean values and also by the standard deviation. So one thing I would like to point out in these FC binding studies is that how we did the fitting of this curve. So if you look at the red trace, that represents the theoretical or the one-to-one Lagmuir fit. So despite optimizing the binding assays very carefully, sometimes these FC receptor binding to MABs display some sort of a complex kinetics, where the dissociation step can sometimes be biphasic. So what this means is that you will see an initial rapid dissociation or one-to-one binding, that is an FcRn binding to the MAP, and that is followed by a secondary high affinity interaction of dimerized FcRn bound to the Fc ligands to dominate the kinetic profile. So the one-to-one kinetic model can be used to fit this more complex interaction when only the fitting of the dissociation step is included in the analysis. Only the first portion of the fitting is included. So it is necessary to truncate the dissociation step by fitting into either the first 5 to 10 seconds. So in this case, we use 10 seconds that allow the initial dissociation rate in the biphasic curve to be captured. So that improves the fitting and provides us with reproducible off rates, which in turn gives us accurate KD values. So this concludes case study 1. The study was performed back in 2019, and now we perform sample testing to this client at least twice a year. And each time it's with fresh FC receptors that we purchase, and then we biotinylate it again, fresh buffers, fresh analytes. However, we also make sure that we have a controlled monoclonal antibody that we always use to make sure our internal comparisons are taken care of. The rate constants has been great thus far. It has been very, very reproducible and within range to what we observed back in 2019. So this suggests the establishment of a reliable and a robust BLI method to evaluate the FC binding studies at Custom Biologics. So with the conclusion of case study 1, now we proceed to case study 2. As you know, monoclonal antibody drugs are complex and heterogeneous mixtures of various variants. Glycosylation of biopharmaceuticals, they show a high degree of heterogeneity, and this N-glycation that happens is most complex and leads to diverse structures in nature due to the different sugar moieties that you see on the slide and possible linkages associated. So the N-glycans have important structural functions. They stabilize the CH2 domain or the F of the FC region of the IgGs, and deglycosylation of these monoclonal antibodies gives rise to thermally less stable molecules and also more susceptible to folding. To unfolding. So MAP typically, the monoclonal antibodies typically exhibit a half-life, body half-life about 10 to 25 days, since they are not usually eliminated by the kidney filtration due to their size, and also about what we talked about earlier, how they escape the degradation in the lysosome through the neonatal receptor recycling mechanism. So there are two major pathways for selective glycoprotein clearance. One by the glycoprotein receptor, that is in the liver, which I'll not be talking about today. And the second is through the macrophage mannose receptor, which is mostly expressed on immune cells such as macrophage. And hence binding of these monoclonal antibodies to the mannose receptor is important to be assessed. So here we are skipping the optimization assets, but briefly here we are using protein L biosensors to immobilize our antibodies via the light chain. We use the same optimization binding assay where we are using parallel biosensors with and without load. In this case, it is an antibody down approach. So we immobilize the monoclonal antibodies at 5 micrograms per ml. And then we also have a parallel biosensor set without a load. And they were all dipped into the MMR or the macrophage mannose receptor at different concentrations. So here we used 5 non-zero concentrations. So one thing to note here is that the association is quite long and the dissociation as well. So even though once you obtain the dose response curves and when they are fitted to the one-to-one model and the dissociation on rates, off rates, KDs are calculated, it's necessary to see that the KD value in this case is very much similar to what we got in the FC gamma R3 study. But the on rate and the off rates here are quite different. Even though the affinity values are the same. So this gives us some sort of a mechanistic or a MOA type information, because it not only gives us the affinity values in general, but also how fast they interact and how fast they dissociate. So here it's a very slow association and it's also a very slow dissociation. So it is necessary to at least have a little bit of curvature and then the dissociation signal to where at least you see a 5% dissociation. That allows the kinetic constants to be calculated accurately and your chi-squared values and to be low as possible and R-squared values to be as high as possible. So in addition, we also moved on to performing a specificity to figure out if there's the specificity of the assay by subjecting the antibody to a deglycosylation. So we used a commercially available kit to deglycosylate the antibody using the endo S2 enzyme. And then we compared the native antibody to the glycosylated antibody and then observed the real binding responses, absolute binding responses. So you can see here that the deglycosylated antibody, when the same concentrations of the MMR are used, so in other words, the loading happened here with the native antibody. Here the loading on the right-hand side, you can see the loading happened with the deglycosylated antibody, and they were dipped into same MMR preparations. But you can see in the graph below that the native antibody bound stronger or bound at a higher rate than the deglycosylated antibody, suggesting the specificity of the assay. So the extent of deglycosylation in this case is not known. We do not know the percentage of deglycosylation, but provided that deglycosylation has actually happened in the presence of the kit, we do see a differential in the binding to MMR. So this is a snapshot how the data looks like. So our task was to compare our originator to 2 different biosimilars. So the 3 MABs, that's the originator, biosimilar 1 and biosimilar 2, were loaded onto protein L biosensors at 5 micrograms per ml. And until a BLI measurable signal of maybe about 1 nanometer was achieved, and then dipped into one preparation of macrophage mannose receptor. So it's the same concentration that we used on the slide, but on the previous slide, but it is in nanomolar concentration, which is important for the calculation of our affinity constants. So as you can see, the responses of the originator biosimilar 1 and biosimilar 2 are very similar to one another, not only in terms of by the responses, but also in terms of the sensograms and also the affinity constants, on rates, off rates as well. So the errors, if you look at, it's more than a log lower than the constants itself. The chi-squared values are lower and R-squared values are much higher. So this concludes case study 2, but I would like to show case study 3 where we could numerically assess the comparability of biosimilars to originators. So in case study 3, we look into angiogenesis inhibitors. So briefly, angiogenesis is the process where new blood vessels are formed from pre-existing one. Abnormal angiogenesis is recognized as a hallmark of diseases such as cancer, and there are pro-angiogenic factors that includes vascular endothelial growth factor or the VEGF family. So in humans and the mammals, VEGF-A, B, C, D, and platelet growth factor or PIGF exists, and they constitute the whole VEGF family of factors. And VEGF-A in particular, it exists as different isoforms such as VEGF-A 121, 145, 165 that we are studying, and others like 189 and 206 as well. So VEGF inhibitors have evolved over time. So if you look at this timeline, initially there was Aptamers or pegatinib, was the first anti-VEGF therapy that was approved for the treatment of AMD that binds selectively to VEGF-A 165, but not to any other VEGF-A molecules. And then there comes in 2005, there was bevacizumab, which has been approved for the treatment of metastatic colorectal cancer, non-small cell cancer, and also glioblastoma multiforme. It is a recombinant humanized monoclonal antibodies that bind to all VEGF-A isoforms. And then there was a derivative of bevacizumab around 2006, which binds to all VEGF-A isoforms much strongly, and that's also called as ranibizumab. And then that was followed by also other fusion proteins that show a higher affinity to VEGF-A molecule as well as VEGF-B and PIGF. So this shows that the importance of different VEGF inhibitors, the role that they play in cancer-associated angiogenesis is actually quite immense. So there's a constant development of different biosimilars to VEGF inhibitors as well. So as a part of the efforts, we were tasked to determine and compare the binding ability of an anti-VEGF monoclonal antibody originator to its biosimilar. So what if we see in slide 24 is how we assess the binding of an originator at different potency levels. So here the optimization I have skipped as well. So to give a brief idea of what we have done here we use AR2G biosensors. So we have used a wide variety of different biosensors throughout the studies. So AR2G biosensors immobilizes the ligand via amine coupling. So we amine couple VEGF-A 165 to the surface of biosensors, and then we dip them into different concentrations of the antibody. So the potency or the relative binding potency, the potency is very important for an antibody molecule to understand how strong it is in functional aspects. So in terms of the 100% potent sample, the concentration range that we use of the antibody was 1800 to 2.5 nanometers. So if you look at the sensogram in the center, that is at 100% potency, the triplicate assessments were performed, the mean responses was recorded, and the standard deviation between replicates was very low. But if you prepare an 80% potent sample where the actual concentration is ranging from 1440 to 2, that is the first sensogram and the first table, The mean responses from triplicate studies were recorded and the standard deviation was recorded as well. And then when you prepare a 120% potent sample, the mean responses from triplicate measurements were recorded. So these mean responses were then used to fit to a 4PL curve to determine the EC50 binding values. So the EC50 binding values were then used using the equation that you see on your right side to understand the relative binding. So here we used only absolute responses to identify to understand the relative binding, but you can also use KD values, the affinity constants as well. So the idea behind performing the potency study was because we wanted to understand the linearity of the assay, how the assay changes when an 80% potent sample or a 120% potent sample is obtained. Whether can you determine the relative binding accurately and precisely. So, and here you can see that the 80% potent sample returned a relative binding of 80.9 and a 120% potent sample returned a relative binding of 120.7. So suggesting that in future, if we get an 80% potent sample or a 120% potent sample, we could actually assign an accurate relative binding value. So then the next task was also to see how an antibody originator from the same company looks like with biosimilars. So here an originator biosimilar 1 and biosimilar 2 was used as the analytes. VEGF-A again was immobilized via amine coupling to AR2G sensors, and triplicate studies were performed. So here you can see that the binding sensograms are very similar to one another, and the mean responses were very similar. Standard deviations were very low as well when the range of concentrations of MABs ranged from 1800 to 2.5 nanomolar. And when these mean responses were plotted again using 4PL curves, you can see that they are very similar to one another. And these curves were then used to determine the binding, the EC50 values, and using the equation on the right, the relative binding was determined. So the biosimilar 1 and biosimilar 2 returned relative binding values of about 89% and 92% respectively, suggesting that the two biosimilars behaved very similar to the originator. With that, we conclude case study 3. So to summarize, the Octet has helped us gather valuable data for a number of clients. However, to obtain reliable and robust binding data, binding assays have to be optimized very carefully. And I hope I have been able to shed some light on that. And these are a few lessons that you can take home. So it is important to determine the best biosensor that is suitable for the binding study. That not only depends on the tag that your ligand comes with, but it also depends on the ligand orientation, the density, to be etc. as well. For example, if you use protein A biosensors to immobilize the MAB molecules for an FC binding study, that might not be ideal because the FC portion has to be left free to bind to the FC receptors. Secondly, it is necessary to optimize the buffer systems to obtain reproducible data and to ensure that non-specific binding is actually absent right at the beginning of the assay. So in my experience, the kinetics buffer from Sartorius has been proven to be the best buffer thus far, and the formulation has helped to reduce this non-specific binding of many of our analytes to the biosensors. So sometimes the NSP, there are also areas that NSP cannot be completely overcome. And in that case, and if the NSP is about non-specific binding is about 20% of the real binding signal, a strategy called double referencing can be used, where you have two sets of parallel biosensors, like what we have done in the establishment assays, where you have the load and the no load biosensors. So the real binding signals are then subtracted from two entities, that is one with the no load biosensors as well as with the reference sample biosensors. That's where the term double referencing comes in. Another approach to make sure your binding event is real and specific is to use a denatured or a mutated analyte, similar to what we performed in case study used to using deglycosylated MAB. The fourth point is use high quality reagents. We often use purified proteins from qualified vendors to avoid issues with either metrics or aggregates. And we usually purchase carrier-free reagents because we do not want BSA being supplemented. Finally, it is always efficient to use the throughput capabilities of the octet to establish assay conditions in a fraction of the time. So multiple assay conditions such as the buffers, loading densities, different analyte concentrations can all be tested in one assay if you have the right planning. So with that, I would like to acknowledge my team at Custom Biologics, from our President CEO, Dr. Dan Marmalak, to the scientific team, admin, and QA team. I'm extremely thankful. I would like to thank our in-house Octa team, composed of Nicole and Dana. And my thanks goes to Austin for creating some of the images for my talk today. Finally, I'm thankful to the Sartorius support teams for always being there to answer my queries and also for help in troubleshooting the assays. The application notes, the technical notes, white papers, and especially the knowledge base at Sartorius from Sartorius has proven to be extremely useful to me and my team to understand how the assays work in general. To that end, great science is at the heart of everything we do at Custom Biologics, and the Octet system has played a role in achieving this goal effectively and quickly. And Custom Biologics is also currently hiring. So if you're interested in joining a passionate group and are excited about the work we do, please feel free to send in your resume to the address on the slide. Thank you.
Right. Thank you very much, Kalhari, for this very nice and informative presentation. We are entering now the live Q&A session. So if you have any question and you haven't submitted it yet, please do so by using the box underneath the presentation window. You put in simply your question and hit the submit button, and we will try to answer as many questions in the next minutes. So for the beginning, we have two questions related to non-specific binding. So first of all, when the map shows non-specific binding, what exactly do they bind to, and how can I prevent or avoid non-specific binding in my experiments?
Sure. Thank you. So thank you for the question. I think it's a great question. And most of my talk was focused on non-specific binding. So the non-specific binding can occur as a result of many factors. So it could be something in the media, it could be BSA, serum proteins, or any charged molecules that's in the assay buffer or in the sample. The non-specific binding that we observed in our case was the MAB, or the monoclonal antibody, binding to the biosensor itself. So in that case, it was the high precision streptavidin biosensors. So the other type of non-specific binding is the binding that we see of the analyte to non-specific areas of the ligand itself. So it's necessary to strategize to overcome these non-specific binding. So non-specific binding blocking agents are there, like skim milk. You can adjust the salt concentration to increase the stringency of the reaction. BSA, Tween 20 are all reagents that you can use as blocking reagents. Something that we have used in the past and which has become very important for us is optimizing the buffers, and also maybe trying out other sensors that are suitable for your platform. So for example, if we tried high precision streptavidin, we can try also protein L or HC. So you can use the array of different biosensors that Sartorius offers and then overcome non-specific binding in that way. So that that was something that was useful to us. And it's important to either use the no-load biosensors to figure out if there's non-specific binding or not, or if you have already tackled it, or alternatively you can use maybe a mutated protein or mutated analyte, deactivated type of analyte also, like what we use for the MMR study, where we use a deglycosylated antibody to understand if your assay is specific and there's no non-specific binding.
Okay, thank you, Kalhari. Another question which came in is, can you a bit explain how the association and dissociation times are determined?
Yeah, so that's a good question as well. So the association step should be run long enough so that there's some sort of a curvature of the data traces, but that should not be too long that the curves tend to flatten out for an extended period of time. So typically 5 to 10 minutes is what is advised for an association step, but that also is reliant on what sort of binding interaction are you looking for. Is it a high affinity interaction? Is it fast kinetics? So in case of fast binding molecules, it should not be longer than 1 to 2 minutes. But if it's a slower association, like what we saw in the MMR, it has to be a little longer. But there should, at the end of the day, it should not be too long association step that there will be an opportunity to weaker non-specific interactions to happen as well. On the other hand, dissociation step should be run long enough so that we see an appropriate level of dissociation. So 5% is what is recommended. And if it is a high affinity interaction, we should definitely run it longer so that there's sufficient time for 5% of the dissociation to take place. That's what we usually follow to get accurate kinetics.
Okay, thank you. And in some of your slides, we saw sensorgrams. What does it mean to have negative binding?
Mm-hmm. Yeah, so negative binding we observe when there are like larger molecules on this biosensor surface that could result in a negative shift rather than a positive shift. Sometimes the negative shift can also happen when there's a conformational change that is happening on the biosensor surface. Has the protein, as a dimer protein, has it dissociated of some sort? So then you would see a negative shift happening as well. Particularly in the case of nickel NTA biosensors, we observe that quite a bit when we use his-tagged ligands. And in that case, to improve the baseline or the stability of the sensor surface, you can also look into a chemical cross-linking approach, like what we do for amine coupling, with amine coupling sensors, so that you can stabilize the loading step a little bit further, and these type of negative shifts are not observed.
Okay. And we have here another question. So what are the affinity ranges you can measure?
Mm-hmm. Yeah, so currently we use the octet red 96 system. So the affinity limits in that case were 1 millimolar to around 10 picomolar in my memory, and the analyte molecular weights should be greater than 150 daltons. So I think if you search for the desired instrument and that associated data sheet, you should be able to find that information relevant to your instrument.
Okay, thank you. We don't have any further questions right now. So please be reminded if you have any question later on, this webinar will be as well available on demand later on. And you can always put in questions when you listen to this on-demand recording. And we will actually receive the questions so that Kalhari, as our presenter, is able to answer these questions as they might occur later on. With that, thank you again, Kalhari, for this great presentation today, for your time you put into the preparation for that and being here today. And I want also thank you, Sartorius, as the sponsor of this talk. And as I already mentioned, this recording will be available on demand, and it is hosted by LabRoots for the entire year. So this means whenever you want to come back or you want to share this with one of your colleagues, you can do this at any time. Share the link, and then everyone is able to watch this later on. Thank you very much for your time today, and have a great rest of the bioprocessing presentation and virtual event. Thank you.